Hello, everybody. Welcome to our webinar on an overview of spectral analysis methods. My name is Yinfen and your presenter for today.
Hi, everyone. A warm welcome also from my side. My name is Kiris, and I'm happy to be your today's moderator.
Hello, my name is Fernando. I will also be your moderator today.
Thank you, Kiris and Fernando. We will start the webinar now, and let me turn off the camera. The intended goals of this webinar are first, to provide an overview of spectral analysis methods, and next is to show how to apply those analysis methods in Analyzer 2, and lastly is to compare the results of applying these methods on an experimental data in order to elucidate the relationship between them. Hopefully, at the end of the webinar, you would be able to understand the differences and similarities between the methods. Based on the obtained knowledge, you will be able to make an informed decision on which method to use in your EEG data analysis. More importantly, with a couple of demonstrations, you will also be familiar to the steps on applying the spectral analysis transformations implemented in Analyzer 2. Now, let's take a look at the contents for today's session. We will begin by revising the origins of EEG oscillations and the classification of these oscillations into different frequency bands. Motivations on why we perform spectral analysis on EEG signal also will be outlined. Next, I will move to the core part of today's webinar. First, Fast Fourier Transform method will be introduced, followed by Wavelet Transform, event-related synchronization or desynchronization, complex demodulation, and Hilbert Transform. For each method, I will provide the theoretical background and a demonstration in Analyzer. In the third session of the webinar, the analysis results of the methods will be compared. Here, the characteristics of each method will be summarized, so you will have a full view on the differences and similarities between the methods. After that, options that are available in Analyzer 2 to export the analysis output will be provided. So, as an estimation, I will need a good 90 minutes to deliver the mentioned contents. Then, we will have the questions and answers session. In the end, I will provide you with some take-home messages. So, let's start the journey. As we know, EEG oscillations arise from the interplay of many neural assemblies through excitatory and inhibitory synapses. What you see here on the very left is the zoomed version of a single pyramidal cell. These pyramidal cells are usually found spatially aligned in the most superficial layers of the brain, and their activity is synchronized, which produces a larger signal that can be measured from the scalp. Electrical signals transmitted along a dendrite creates charges which acts as a battery, known as primary currents, with a positive polarity, where the current is leaving from the cell body, and a negative polarity, where the current is returning to the cortical surface. The postsynaptic potentials of many of these pyramidal neurons, modeled as electrical dipoles, highly contribute to the signal measured by EEG. These major oscillatory activities can have a repetitive pattern, the so-called rhythmic activity, or evolve over time, the so-called transients. Moreover, oscillations can be classified as spontaneous, evoked, or induced. The rhythmic oscillatory activity of an EEG signal can be categorized into different frequency bands. In EEG research, there's a nomenclature of frequency bands which is summarized in this table, from the lowest delta to the highest gamma frequency, or from the slowest to the fastest wave when observed from the morphology perspective. The definition of the borders of the frequency bands vary from publication to publication. These brainwaves differ in shape from one individual to another, depending on the functional status of the brain. So, the brain state of the individual makes certain frequencies more dominant. For example, the well-known alpha wave rhythm predominantly originates during wakeful relaxation with closed eyes. By investigating the frequency content of these oscillations, we can infer on different mental states. For example, when you are awake and calm, your brain will generate more alpha waves. If you are highly focused to what I'm talking and actively engaged in the webinar, beta or gamma oscillations will be induced in your brain. But if I bore you, your brainwave will be dominant with theta waves and maybe even delta waves if you fall asleep. But I hope this is not the case. Furthermore, when looking at EEG in terms of oscillations, we can see how they synchronize across brain regions, and this can tell us about the connectivity throughout the brain. You can learn more about this in our webinar, Phase and Connectivity Analysis. Now, the important question is how to extract the frequency content of the EEG signal. The answer is by performing spectral analysis methods that decompose the EEG signal into its constituent frequency components. The spectral analysis methods are further divided into frequency and time frequency domains. We will cover both in this webinar. Let's start with the FFT. First of all, I would like to mention that we have a dedicated webinar just for FFT, where you can grasp the detailed information and mathematical descriptions. I will attach the webinar channel link at the end of the talk today so that you can watch it at your convenient time. In the following slides, I will give you an overview of the method so that you will get a general idea on the FFT. Let's begin. So, what is Fourier transform? According to Joseph Fourier, any signal can be represented as a summation of simple sinusoids, namely sines and cosines. Each of these sinusoids are defined by their frequency. How fast is the oscillation, measured in cycles per second or hertz? Amplitude or power, the strength of the signal, and phase, that tells us the offset of the oscillations from its starting point, measured in radians or degrees. In other words, any time domain signal, for example this one, can be decomposed into its constituent frequency components. As you can see, the original signal is composed of 20, 30, and 40 Hz frequency components. The next question is, how should we calculate the amplitude at those frequencies? As you might have guessed, the answer is to use Fourier transform. Let's say we have a signal which is composed of stationary 1, 8, and 30 Hz oscillations. With Fourier transform, what we do is we divide the data into epochs, for example of 1 second length, and then analyze the frequency content of these epochs. When decomposing, for example, this section of the data with Fourier transform, we probe it with templates of different sinusoidal functions and determine how strongly different frequencies are represented in this data stretch. This is done by multiplying the EEG signal with a complex sinusoidal function, which tells us how similar are the two signals. Whenever the EEG signal and the sinusoidal function are similar, they will multiply and sum to a large coefficient. And whenever the EEG signal and the sinusoidal function are dissimilar, they will multiply and sum to a small coefficient. In this specific case, the Fourier transform finds a strong representation of the frequencies 1, 8, and 30 Hz because they make up this simulated data and would show peaks at these frequencies in the resulting power spectrum. Due to complex sinusoids, the result of the computation will be a complex number per frequency, which are called Fourier coefficients. A complex number means the number is represented as A plus IB, having real and imaginary part. So what can we do with these complex coefficients? We can make use of them to compute spectral amplitude, power, and phase by using the following equations. Another important point of Fourier transform is that the frequency range that we can evaluate goes from 0 to half of the sampling rate, the so-called Nyquist frequency. For example, if we have a sampling rate of 512 Hz, we will be able to evaluate frequencies only up to the Nyquist frequency, which is from 0 to 256 Hz. There is an important assumption to remember prior to applying Fourier transform. Fourier transform is an adequate tool to analyze stationary signals. Because the result we obtain from the Fourier transform is easier to interpret if the input signal is stationary as compared to an input signal which is non-stationary. So, stationarity is the keyword in this topic. However, EEG signals are non-stationary by nature. That means the morphology of the signal varies over time. Therefore, it is important to apply Fourier transform to those signals that can be assumed as stationary or quasi-stationary. Quasi-stationary signals in the sense that the spectral properties vary slowly with respect to the carrier frequency or the main frequency, so that we can still get meaningful result. So, when we apply Fourier transform, we need to first extract portions of data that can be considered as quasi-stationary. Now, we have covered the theoretical background of FFT. Let's move to its implementation in Analyzer 2. In Analyzer, we offer the Fast Fourier Transform algorithm, which is an algorithm for fast calculation of discrete Fourier transform. For the demonstration, we are going to use the data acquired from a resting state paradigm, where participants were asked to keep their eyes open and closed. In this demo, we will show you how to perform FFT analysis in Analyzer and validate the already known hypothesis that alpha activity increases when the eyes are closed, resulting in a larger spectral power between the alpha range. All right, let me open Analyzer. The data I will use in this demo is named as resting state data that I show here, that consists of 3 blocks, eyes open, and 3 blocks, eyes closed data. Let me expand the history tree so that you can see the whole processing pipeline that is already prepared. The data was recorded with a sampling rate of 1000 Hz, and FCZ was used as the online reference. It was bandpass filtered artifacts were rejected and the ocular correction ICA was performed, it is then downsampled from 1000 Hz to 512 Hz, which is a power of 2, to avoid zero padding and obtain the desired frequency resolution. The data were segmented twice. So the first segmentation was to extract portions of data that can be considered quasi-stationary. So here the data has been segmented based on eyes open markers. This gives a total of 3 segments. The second segmentation was performed on the first segmentation nodes to obtain equal-sized segments. In this case, it is 2 seconds long. After that, FFT was applied on the segmented data, and the transformation can be found under Transformation, under Frequency and Component Analysis, FFT. So let me open the FFT dialog page and take a look. I can do it by clicking either Edit Parameters or Reprocess, or Edit Parameters or Copy. So I will click on the Edit Parameters, Copy. The FFT transformation has only one dialog page. The very first group of the FFT is about defining the frequency resolution. As you can see here, by default, Analyzer shows us already the maximum resolution that we can achieve with the input data that we have prepared. That is 0.5 Hz. The second group is the possible output options. Since we are interested in the spectral power, I have selected power and the full spectrum option to obtain a one-sided spectrum. The third group is about data windowing. We are using a Henning window with 10% window length. This means 5% of the signal is attenuated from the left and the right side prior to applying FFT. Since we are not going to recalculate the FFT, I will click Cancel. To improve the signal-to-noise ratio, all resulting 3 spectrums were averaged. So this is the resulting average spectrum. Let me select an oscillator channel to look closer by double-clicking example channel offset. Let me upscale the amplitude. We can see a relatively high spectral power. Sorry, let me click on the average power. So let me upscale again the amplitude. So we can see relative high spectral power in the alpha range, which is shown in green. So all the steps were replicated to the eyes closed data. Now we can compare the spectrum of eyes closed to the eyes open. One easiest way to compare two conditions is to create an overlay. Let me drag the average power node for the eyes closed condition and overlay it on the eyes open data. So the red line is the spectrum of the eyes closed condition. We can see a tremendous differences between the alpha range. As expected, the spectral power in this alpha band is higher for the eyes closed condition as compared to the eyes open condition. With this, I will end the demonstration and go back to the slides. Let's summarize what we have learned so far. FFT maps time domain signals to frequency domain representation. It tells how much of a signal is contained in the total period of observation. The mapping is done by computing the similarity between the signal and the sine waves of different frequencies. The output of FFT is a complex number in which the power and phase can be extracted. The resulting power spectrum represents the activity of neural oscillators per frequency, allowing the comparison between conditions or tasks or bands. It can be used as long as the underlying oscillatory patterns are quasi-stationary. An example will be performing spectral analysis of the alpha activity during eyes closed, eyes open paradigm. Now we will dive into the time frequency analysis topic. Recall that one assumption of the FFT is that the data are stationary, which means that the statistics of the data, including the mean, variance, and frequency structure, do not change over time. What if the signal is non-stationary, but if the frequency content change transiently? What will happen we apply FFT on this kind of signal? Let's take a look. This signal is composed of 1, 8, and 30 Hz oscillations, but their amplitudes are modulated by lower frequencies. So these oscillations are not stationary but transient. The problem is that with FFT, we can only look at the frequency content of sections of data. And we will receive a very different frequency spectrum in this section than in this section of data. So with FFT, we cannot track the changes dynamically. To investigate such spectral dynamics across time information is required in addition to frequency. So we would like to have an analysis technique that informs about the frequency content and at the same time preserve temporal information. This is exactly what time frequency analysis offers. There are many time frequency analysis methods, but we will focus on the highlighted ones in this webinar. Let's start with Wavelet Transform. This method is no doubt a widely used time frequency decomposition method in various areas of EEG signal or EEG research. Same as FFT, we have also a dedicated webinar video for Wavelet Transform, where you can get the detailed information and mathematical descriptions. So in the following slides, I will give you a general overview of the method. Like FFT, the Wavelet Transform quantifies the similarity between the signal and the wavelet of different center frequencies. But what is a wavelet? A wavelet is a short sinusoidal wave of one center frequency multiplied by a Gaussian function to preserve temporal information. There are different kinds of wavelets, but the most commonly used ones in EEG studies are complex mode wavelets, which contains a real and an imaginary part. How does wavelet transform work? Let's assume we have a transient signal and imagine we center a wavelet on a specific time point on the signal. We then convolve the signal with the wavelet. During this process, the wavelet is matched with a correspondingly long stretch of data, and the similarity of this data stretch and the wavelet is calculated. The result is one value, shown in green, for the time point the wavelet was centered on, called a wavelet coefficient, that tells us how much the data matches the wavelet at this place. Let's, for example, say this wavelet has a frequency of 8 Hz. Then the result tells us how strongly 8 Hz oscillations are represented in the data. A high value would mean a high similarity of data and the wavelet, telling us that 8 Hz oscillations are strongly present at this moment. We apply this calculation for every time point of the data. What we receive is the continuous time cost of the power of 8 Hz oscillations in the signal. Usually, we perform wavelet calculation for several frequency layers. As we said, this signal is composed of 1, 8, and 30 Hz oscillations of fluctuating amplitudes. If we plot the signal with a 1 Hz wavelet, we will find increasing 1 Hz content towards the end, depicted in hot colors. We can do the same for 8 Hz and 30 Hz, and in fact for a whole number of frequency layers in between. What we receive is a heat map of frequency by time, where hot colors indicate a similarity and cold colors indicate dissimilarity between the signal and the wavelet. This is called the wavelet power spectrum. We can interpolate across time and frequency points to make the wavelet power spectrum look smoother. We have time and frequency information from the wavelet analysis, but can we get simultaneously high frequency and temporal resolutions? The answer is no. If we want to increase the frequency resolution of the wavelet analysis, we will necessarily decrease its time frequency resolution and vice versa. For example, with a relatively long 10 Hz wavelet that covers more data points, we can localize the power of 10 Hz oscillations precisely and achieve a high frequency resolution. However, due to the influence of many neighboring time points in calculating the coefficients, a long wavelet yields a low time resolution. In contrast, if we instead use a shorter 40 Hz wavelet, a broader range of frequencies will contribute to the resulting wavelet coefficients. This means the shorter 40 Hz wavelet would provide higher temporal but lower frequency resolution. Now, how we can modulate this time-frequency trade-off? For this, we can use the modulate parameter C. This parameter determines how many cycles are used for the wavelet. I'm showing here 3 wavelets, each with a center frequency of 8 Hz, but they differ in their C parameter, so in their number of cycles. As you can see, this number of cycles influences the length of the wavelet. Let's say I have a modulated 8 Hz signal. If I perform 3 wavelet analysis, each time using a different C parameter, you can nicely see how this affects the resulting time frequency spectrum. Using more cycles will increase the frequency resolution, meaning the frequency band gets narrower. At the same time, the signal gets more spread over time, so the time resolutions gets lower. Due to complex modulate wavelets, the result of the computation is a complex number per frequency and per time point, which are called wavelet coefficients. Same as FFT, we can compute the amplitude, power, and phase from the wavelet coefficients by the following equations. Now it is the time we move to the demo part. The purpose of the demo is to show you how to compute, evoke, and induce power using wavelet transformations in Analyzer 2. For the demonstration, we are going to use the data acquired from a visual evoke paradigm. During the task, participants were told to focus on a fixation cross on the center of the screen. Then a white dot of 10 mm diameter was presented at random locations in one quadrant of the screen. This task works as a nice example because it underlines what different results we can receive if we look at evoked power, which is time and phase law, or induced power, which is non-phase law. First, let us consider the evoked power. We know that presenting a visual stimulus will elicit a response. If we look at the time domain signal, we reliably find a visually evoked potential around 150 milliseconds after the stimulus onset. This potential stems from an increased phase locking in the alpha range. This means the neural activity gets resynchronized by the stimulus onset in a 10 Hz rhythm. If we look at the power spectrum of purely evoked activity, we will expect an increase in alpha power shortly after the stimulus onset. The picture will be different if we look at the induced or whole power spectrum. Even though the alpha rhythm resynchronizes, we find that its overall amplitude decreases when we display visual stimuli. So alpha oscillations get suppressed or blocked in the visual cortex when paying attention to the visual stimuli. So when analyzing total or induced power, we would expect a decrease in alpha power following the stimulus onset. Let's go to analyze it now. Like the FFT demo, the whole processing pipeline is already prepared. And I will show you and explain you the important settings of the wavelet transformation that are used. Let me minimize the resting state history tree and expand the alpha blocking data that we are going to use in this demo. The data contains recording of 30 scalp channels. It was recorded at 1000 Hz sampling rate and was originally referenced to the right mastode. It was pre-processed following the normal pre-processing steps, and it has been segmented around the stimulus markers S1 with a time range from 1 second pre-stimulus and 2 seconds post-stimulus onset. To obtain the induced power, I have first computed the total power on the segmentation node and subtracted the evoked power from the total power to get the induced power. The power was computed by using the wavelet transformations, which can be found under Transformation, Frequency and Component Analysis group, Wavelets. So let's take a look at the wavelet dialog. I can also do it by clicking on the Edit Parameters and or Copy. So the type of the transform that we used is the continuous wavelet transform, which is also by default is chosen here. So let me proceed by clicking OK. The wavelet transformation has two dialog pages. This is the first page. Under the wavelet group, the complex mode wavelet is selected and also is by default. It is selected. The frequency range from 1 to 40 with the logarithmic steps of 40 frequency steps was specified, and the mode parameter was set as 5 cycles. Those are also the default values, and I will keep these values here. And here you can see the resulting wavelets depicted in the time domain, where the black color is the real part of the wavelet and the red color is the imaginary part of the wavelet. To go to the second page, I will click to the next. For output values group, the spectral power option is selected, and I don't select the normalization option here in this demo. If you are interested to know more about this option, you can refer to the mentioned dedicated webinar. Since I'm not going to reprocess the node, so I will click Cancel. Let's check the results, which are the Evoked and also the induced power. Let me close the window first. And let me arrange it bottom and top so that we can see the differences. So since the effects are most prominent over the visual cortex, let me select the occipital electrodes, for example, channel P03 to have a closer look. So the upper figure here is the evoked power result, which is time and phase lock power. As expected, there is an increase in the alpha power shortly after the stimulus onset. The lower figure here is the induced power result that is not strictly time and phase locked to the stimulus. We can see an overall reduction in the alpha power. This is the predicted alpha blocking that occurs when the stimulus appears on the screen. So the evoked and induced results are different. And this is in line with the hypothesis. With this, I will end the demonstration of wavelet analysis and go back to the slides. Let's summarize what we have learned about wavelet transform. The wavelet transform is a time-resolved frequency decomposition method used for localizing changes in the frequency characteristics of a signal over time. It is done by quantifying the similarity between the signal and the wavelet for every time point of the signal for wavelets of different center frequencies. The resulting wavelet coefficients is a complex number at each time point and each frequency. From these coefficients, amplitude and phase can be computed. It can be used to perform evoked power and induced power analysis in an event-related paradigm. Now, I would like to present another method that can also be used to investigate the spectral dynamics of an event-related signal. The method is called event-related synchronization or desynchronization, or in short, ERS-ELD. So what is ERS-ELD? Quoted from the review paper on the method by Grumman and Puscheder in year 2006, the ERS is defined as the proportional power decrease or increase in relation to the baseline activity during a specific reference interval that is usually selected to be several seconds before the event onset. This tells us that in computing ERS-ELD, we are checking obviously signal power. This can be in a specific frequency band, and this is actually very similar to what we did with FFT. However, we are not investigating absolute power, but relative power as we quantify the decrease or increase from some baseline activity. And the whole process is related to some event. This is very similar to ERPs, where we also look for decreases or increases in amplitude. We also relate this to a baseline as we baseline correct the data to center our ERP around 0. And of course, there is an event to which the whole activity is logged. Thus, ERS-ELD is thought to as an extension of the ERP method into spectral analysis. The result of ERS-ELD is a time-resolved instantaneous band power of a specific frequency band, which means that we keep time resolution, however, to the cost of being constrained to a specific frequency range. It is typically expressed as relative to the mean or median value between a reference interval, for example, percent change. When the results are of positive values, it is referred to an increase or ERS. When the results are of negative values, it is referred to a decrease or ERD, which is shown in this figure. So, how to compute ERS-ELD? You will see that computations involved are very simple opposed to other spectral analysis methods, for example, the Wavelet Transform. There are 4 main steps involved. First, the segmented data is bandpass filtered to extract a frequency band of interest. We do this as we want to know the power changes that are specific to some frequencies. Please note, this is a necessary step before applying ERS-LD. Then, the power of the signal is computed. There are 2 methods for the power calculation. The first is the original one, which is called power method, and does nothing but squaring. This leads to a preserved evoked and induced activities. We could not disentangle for one frequency band if an effect is caused by one or the other activity. That is why, additionally, the intertrivariance method has been suggested, where the evoked activity is subtracted first before squaring the signal. This will only preserve induced activity. So that means we could actually compute ERS-ELD with both methods and then compare the results. If they are the same, we could conclude that the effect is solely based on induced activity. After that, trials are averaged as in ERPs. The result is the instantaneous power or intertrivariance. So the relative aspect of ERS-LD is still missing. To compute this relative measure, for example, in percentage, the formula below is used. First, the mean of the data points between the reference interval will be subtracted. At every data point of the average power, the original paper suggested that the median can be used alternatively as well. Then, it is divided by the mean of the reference and finally multiplied with 100%. In other words, the power of the signal is expressed with respect to the reference, whereby the reference is defined to be 0. In Analyzer, we call the corresponding options of the 2 power calculation methods instantaneous power and intertrivariance. There is an additional important feature about the method which might be important to know, though it is an optional step. As we often do in ERPs, when we extract the amplitude information of a peak or time range, we average across data points. This gives us a more reliable estimate of the result that is not biased that much by fast fluctuations. In ERS-LD, this is usually implemented via a moving average. Regarding the window length used for calculating the moving average, there are 2 recommendations. The authors of the aforementioned review paper recommend using a longer average time across data points if there are very few trials, while a shorter average interval should be sufficient if there is a lot of trials. This is because the effect of both averages across time and across trials leads to a similar result. Another study suggests that half a period of the frequency of interest would be an optimal window for averaging. How is the moving average performed? Assuming this is an ERS-LD curve, the operation involved is taking each data point of ERS-LD and replace its value with a mean value from a window around that point. Eventually, the data is smooth. As a consequence, we get a more reliable power estimate that is less biased by outliers. However, note that there will be some artifacts at the edges of the segment because of missing values outside of the segment borders. This effect disappears after half of the smoothing window. The theoretical background of ERS-LD has been covered. Now, let's move to its implementation in Analyzer. Let me close the window. We will use the same data set as in the wavelet demo, the alpha blocking data. Unlike the previous two demos, we will compute the ERS-LD. So recall that the ERS-LD is computed on the segmented data. So our starting point is the segmentation node here. As a prerequisite for computing ERS-LD, we will need to first filter the data using the bandpass filter to extract the frequency of interest. So let me call the IIL filters transformation, which is located under transformation, artifact rejection or reduction group data filtering, IIL filters. For the alpha band, we want to isolate frequency between 8 and 12 Hertz. So let me enter 8 Hertz as the low cutoff and 12 Hertz as the high cutoff. And both of the frequencies I will use the order of 8. The notch filter is not needed here since it has been applied in the pre-processing step. So I will click OK to finish the step. This will generate a filters node. To keep track of the processing steps, I will rename the node to a telling name. So let me rename it, for example, to filters 8 to 12 Hertz. Now, on top of these filter nodes, we are ready to call the ERS-LD transformation. It is located under Transformation, Frequency, and Component Analysis group. The dialog page of ERS-LD consists of 2 pages. Let's start with the first one. So there is a bandpass filter group here. In principle, we can also use the filter here as the filter is the same that we offer in IIL filters transformation, only that the order is hard-coded to 8 order. The reason I did it before, it's because I would like to have a filter node that can be used later when I carry out the demonstrations for complex demodulation and Hilbert transform. But of course, you can use this filter in your analysis pipeline. Since I have filtered the signal, I will disable the option here. In the method group, you can switch between the 2 ways of power calculations, power or variance. Let's select the instantaneous power method for now. In the smoothing group, you can enable and define the moving average that will take the mean across multiple data points. We learned that there are 2 recommendations. For now, let's use half a period of our central frequency, 10 Hertz. This would be 50 milliseconds, which is also the default value. Finally, In the segment selection pane, you can either select all segments or make sub-selection. These are the same options as in the average transformation. We select full range, so that means all 97 segments will be used. Let me click Next, and this will jump to the second page of the dialog box. There is the first group, the output values group, giving the option to use logarithmic scale instead of keeping the scaling natural to our power values. Let's skip this option for now. Then there is the normalization group and the reference interval group, which have to be considered together. Let me enable the relative values. For example, percent change here, so they are the default values, and I will use this option as well. And also, I will also let the reference as the mean that we will use. As for the reference interval, let's use minus 700 milliseconds to minus 300 milliseconds. This is a very long window, and we stay away from the segment border and the stimulation time where we could expect deviations from a baseline. Lastly, if you want to perform a sign test to have an idea of where significant power increases or decreases occurs, you may enable the option Data Set for Significance in this statistical data group, but I will not select this option for now. Okay, we are all set now. I will click Finish to execute the transformation. Let me rename the node to ERS ELD power, since we use the power method. Let's take a look at the results of channel P03 again. Let me downscale the amplitude so that we can have a better look. So negative values are scaled downward. Therefore, downward deviations means ERD or power decreases. As you can see, there is not much happening before the stimulus, which the value shows fluctuate around 0 values. However, there's a substantial negative deviation after the stimulus onset. This is the alpha power reduction that we are looking for. And this is very much in line with our initial assumption regarding the alpha blocking. So now let's go back to the slides. Let's summarize what we have learned so far about ERS-ELD. ERS-ELD is the extension of the ERP methodology into the spectral content of a signal. It is done by filtering the signal with a bandpass filter to extract the frequency band of interest, computing the power, and averaging the segments. The result is a time-resolved instantaneous band power of a specific frequency band. It can be expressed as relative power or variance change with respect to a reference interval. Same as wavelet analysis, ERS-LD can be used in an event-related paradigm to investigate non-phase lock activities. Now I'm going to introduce another spectral analysis method that can also be used to investigate transient changes of oscillatory EEG signals. It is called the complex demodulation method. Are you motivated to learn more about it? Let's start. Complex demodulation is a method to reconstruct the instantaneous power and phase of signals. Instantaneous power helps us to track the time evolution of transient changes in the signal energy, while the instantaneous phase can reflect subtle transient changes in the carrier frequency of narrowband oscillatory signals. Like wavelet analysis and ERS-ELD, complex demodulation can be used to study phase lock and non-phase lock activities in an event-related paradigm. As you can see, unlike wavelet analysis, but same to ERS-LD, the result is also a time-resolved signal, which is the envelope of the instantaneous amplitude or power curve of the carrier frequency. So how is the complex demodulation computed? The method is really simple and it involves only two main steps: down modulation and low-pass filtering. To illustrate this, let's assume we have a frequency spectrum of a signal as shown here, with the upper frequency border and lower frequency border denoted as FU and FL respectively. In down modulation, the frequency spectrum of the input signal is shifted towards the origin by the quantity of F node, which is also called as demodulating or carrier frequency. The frequency shift in frequency domain is actually equivalent to multiplying the signal by a complex exponential at the carrier frequency in time domain. So the complex has nothing to do with complexity. It is merely a reference to the use of complex exponentials, which makes the mathematical descriptions more concise. Due to the shift, the frequency of interest, that is the carrier frequency component, is now located at the origin, which is the zero frequency. Other frequencies appear as higher range and can be isolated by applying a low-pass filter. The cutoff frequency is obtained by the following equation, which is the half of the spectral bandwidth. For example, if the upper frequency border is 12 Hz and the lower frequency border is 8 Hz, the cutoff frequency will be 2 Hz. There are prerequisites that we might need to take into considerations when performing complex demodulation. First, the carrier frequency should be known a priori. Ideally, it should be the center frequency of the band of interest. Please note that the extracted instantaneous amplitude or power and phase can only be interpreted if the feature of interest in the input signal is well localized in a narrow band frequency. Let me show you what it means. In this figure, complex demodulation was applied to a narrow band signal from 8 to 12 Hz. As we can see, the estimated amplitude envelope in red here, it's well visualized as a smooth curve outlining the local maxima of the signal. Of course, we also observe a little bit reduction of the values due to low-pass filtering. On the other hand, if complex demodulation is applied to a broadband signal consisting of 0.1 to 40 Hz, the resulting amplitude envelope is not so easy to be interpreted compared to the results of the narrowband signal. Although the resulting envelope is still visualized, but the troughs are not well represented. They are distorted by low frequency components. Therefore, a bandpass filtering around the carrier frequency is required before performing complex demodulation. As an additional remark, if you aim for a smooth amplitude or power curve, it is therefore recommended to use a rather narrow frequency band, which means the small difference between the upper and the lower limit, and thus a small cutoff value. Now we are equipped with the knowledge of the complex demodulation method. Let's move to its implementation in Analyzer. So we will use the same alpha blocking dataset for this demo. Remember that to get an interpretable result, the input signal should be a narrowband signal. And actually, we have already prepared this signal in the demo of ERS-LD, which is this filters node. So let me close all the windows. So this will be our starting point for the complex demodulation method. Let me call the complex demodulation transformation on top of this node. So this complex demodulation can be found also under transformation, under frequency and component analysis group, complex demodulation. You can see that the dialog page of this complex demodulation is very simple, and it only consists of one dialog page. In the frequency range group, I will use the same parameter setting as in ERS-LD, so I will enter 10 here as a 10 Hz as a central frequency, which is also the default value. And as spectral bandwidth, I will enter 4 Hz so that we will have the frequency border from 8 to 12 Hz, which is the alpha band. Meanwhile, the spectral bandwidth information will be used to determine the cutoff frequency of the low-pass filter. In this case, the cutoff frequency is calculated as 2 Hz. Same as ERS-LD, the II alpha Butterworth low-pass filter here has been hard-coded as 8 order. Under the output group, for now, the power option will be selected, which is also the default value, to be consistent to the ERS-ELD node. So let me click OK and execute the transformation. I will rename the node to complex demodulation and power. Since the output option was choose as power, so on top of this node, I will apply the average, which is average transformation, which can be found under transformations, segment analysis functions. And I will use full range of the segments. So let me rename the node as average CD power. CD here stands for complex demodulation. So now let's take a look at the result. So again, I will select channel PO3 to take a closer look. Let me scale up a little bit the amplitude so that we can have a better visualization. So this is the result from complex demodulation on the signals that with 8 to 12 Hz. The result is actually the estimated power envelope at 10 Hz, which is the carrier frequency. So we can see that there is a negative going wave, which means a decrease in terms of power right after the presentation of the stimuli. So this is the alpha power reduction that we are looking for. Now I will go back to the slides. Let's summarize what we have learned about complex demodulation. Complex demodulation is one of the methods to extract the instantaneous power and phase of signals. It is done by down-modulating and low-pass filtering the input signal to isolate the modulated signal at the carrier frequency from others. The result of complex demodulation is a time-resolved signal of instantaneous power at the carrier frequency. Like Wavelet Transform and ERSLD, it can be used in an event-related paradigm to investigate phase lock and non-phase lock activities. Complex demodulation is not the only method to extract instantaneous amplitude and phase of a signal. Now I'm going to introduce you another method that is also carry out the same function. This method is called the Hilbert Transform. Perhaps you will ask, is there a Hilbert transformation in Analyzer? Indeed, there is no such module in Analyzer. But the good news is we can still perform Hilbert Transform if we want to. This is done by making use of the Wavelet transformation. I will show you how to achieve it. Now, without further ado, let's review the theory of Hilbert Transform. Like complex demodulation, Hilbert Transform is a widely used signal processing method to retrieve instantaneous amplitude or power and phase of an oscillatory signal. The idea behind the Hilbert transform is rather simple. It simply transforms a real value signal into a complex signal, which is called an analytic signal. There are 3 steps involved in computing Hilbert transform. First, the FFT of the input signal is calculated. Let's say the input signal is a 10 Hz signal with an amplitude of 10 microvolt. The frequency spectrum will look like this, and it is called the half spectrum. As we know, a real signal has an identical positive and negative frequency components. Here we can see that each of them having the amplitude of 5 microvolt. The negative frequency component is redundant and thus can be eliminated with no loss of information. So in the second step, to keep the signal energy, these redundant negative frequencies will be folded over to the positive frequency values by computing its full spectrum. What we get is the same frequency component at the same frequency band with an amplitude that's exactly the same as the amplitude of the original signal. Lastly, in order to be back to time domain, the inverse FFT is performed. The final output of the Hilbert transform is again a time-resolved amplitude or power envelope of the frequency band of interest. For example, when we apply a Hilbert transform to a signal consisting of 8 to 12 Hz, as shown in this figure, what we will get is the amplitude or power envelope of the frequency range of 8 to 12 Hz. The same prerequisite for complex demodulation apply to Hilbert transform. So the frequency band of interest should be known a priori. And in order to get a result that is reasonable and easier to interpret, it is highly recommended to apply a bandpass filter to the input signal before applying the Hilbert transform. To show the importance of this narrowband property in applying Hilbert transform, let's take a look at the figures below. The left figure shows the result of applying Hilbert transform to a narrowband signal. As expected, the calculated amplitude envelope is very well visualized as a smooth curve outlining the local maxima of the signal. While in the case of broadband signal, although the resulting amplitude envelope is visualizable, it is difficult to be interpreted. Therefore, Hilbert transform shouldn't be applied to a narrowband signal. Now, I will show you how to use the MATLAB transformation to perform Hilbert transform. The MATLAB transformation is designed as a bridge between Analyzer and MATLAB. That means you can transfer data to MATLAB, perform further analysis in MATLAB, either using built-in functions or custom MATLAB scripts, and then re-import the data to keep working in Analyzer. Now let's go to Analyzer. Let me close all the windows first. So the same alpha blocking data will be used in this demo. Let me activate the filters data. So we will start again from this narrowband signal. And then I will call MATLAB transformation. It is located under transformation, others group, and MATLAB. Please note that in order to use the transformation, it is required to have a valid license for MATLAB installed on your computer. So MATLAB transformation has 3 dialog pages. So this is the first page. Here we have the options to calculate the data when the node is created or on request. If the calculate data on request option is selected, the computation will be done only for the current visualization window. For now, I will select the option calculate data on creation of node, which is also the default option. Besides that, we can also choose either to raise the MATLAB desktop application or simply enter the MATLAB code into the textbook here to be executed. One important note to the option to raise the MATLAB window, if it is selected, the data export will be terminated by a message window, which must be confirmed in order to return to Analyzer. So this is not recommended if you want to save that step as a history template for automation purpose. I will not select this option for now. In this textbook, as I said, you can enter all the MATLAB code required to process the EEG data. Let me copy and paste the MATLAB code that I already prepared. So we have 2 lines of code here. The first line is basically tells us that we are going to use the Hilbert function from MATLAB to compute the Hilbert transform on the EEG data. And the ABS function is to calculate or to compute the amplitude of the transform signal. EEG data is the variable name used by Analyzer for storing the exported EEG data in a MATLAB matrix. So the expression in the second line here basically is to compute the power of the transform data, which is the amplitude square. So I will click next to the second page. I will not select any export options here. So here we can see that Analyzer offers a very nice additional feature that is to interact with EEG Lab, an open source signal processing environment for electrophysiological signals running on MATLAB. As you can see here, you can export the data in EEG Lab format and also to raise EEG Lab if you intend to. I don't select any options here in this demo as we are neither exporting data to EEG Lab nor using the EEG Lab. So I will click next to the third dialog page. Here you can choose to export all channels or only certain desired channels, I will choose the all channels, and this is also the default option. Then I will click Finish to execute the transformation. The processing of the code is carried out in the background, and a note. The processing is in the background. Okay, and we will see a progress bar showing that progress. So a node named Wavelet Transformation is generated. So I will rename the node to Wavelet Transformation. Since we use Hilbert, so I will put Hilbert, and the output option is the power, so the power that we calculated. So on top of this resultant node, let's compute the same like I did in complex demodulation, that apply an average transformation on it. So let me rename the node to average HT power. So HT here stands for Hilbert Transform. So now we can examining the output. So I will again choose the PO3 channel to take a look. Let me upscale the amplitude. So this is the result of Hilbert Transform. We can see that the signal morphology looks very similar to ERS, ERD, and complex demodulation. This is also a reduction of power at the similar time range. With this, I will end the demonstration and go back to the slides. Let's summarize what we have learned about Hilbert Transform. Hilbert Transform is one of the methods to extract the instantaneous power and phase of signals. It is done by taking the FFT of the input signal, removing the negative frequencies, doubling the positive frequencies, and performing the inverse FFT. The result of Hilbert Transform is a time-resolved signal of instantaneous power of the frequency band of interest. Like Wavelet Transform, ERS, ERD, and complex demodulation, it can be used in an event-related paradigm to investigate phase lock and non-phase lock activities. Now, I would say we arrive at the most exciting part of the webinar. So far, we have discussed 4 different time frequency analysis methods that includes wavelets, ERS, ERD, complex demodulation, and Hilbert Transform. I'm sure you are curious to know if they are different from each other, and particularly which method you should use in solving your research questions. To find out the answers, one thing we can do is to compare the analysis results. Let me switch to Analyzer and show you this comparison. So let me minimize the alpha blocking data history tree. For comparison purpose, I have also prepared a history tree named as comparison of analysis method. Let me expand the history tree. So far, we have the percent change data already for ERS, ERD. For comparison purpose, other methods need to have the percent change data as well. This can be done easily by utilizing the percent change solution. This solution can be found under Solution and Transform, under Transform percent change. This solution is a part of all Analyzer solution package. If you don't have it yet, you may download it from our website. So I have applied the percent change solution to the complex demodulation result and also the Hilbert Transform results and named them as percent change CD power and percent change ht power respectively. For wavelet transform results, there is an additional step required before computing the percent change. So recall that the wavelet analysis that we have carried out is from 1 to 40 Hertz and with 40 frequency steps. But what we want to compare here with other methods is the frequency of interest of 10 Hertz. So in this case, we will need to extract this time course information of 10 Hertz from the whole wavelet analysis result. Let me show you the PO3, what I meant. So as what I said, we did the wavelet analysis from 1 to 40 Hertz with 40 frequency steps. So we want to compare only on this 10 Hertz layer, which is we need to extract out this time course information. So what we can do is we can apply the wavelet extraction transformation that can be found under transformation, wavelet extraction transformation. So this is what I have done here. And on this node, the percent change solution was applied on it to extract out the 10 Hertz time course information. So now we are ready to compare all of them. So let me overlay all the results. So this one is the percent change for wavelet for 10 Hertz, and I will overlay the ERSLD results and also the percent change complex demodulation and also percent change Hilbert transform. So let us take a look at the channel PO3. So this is the comparison results of all the 4 analysis methods. So we can see that although the output of 4 different time frequency analysis methods are not exactly the same, they differ in terms of the values, especially at the borders of the segments. But they all exhibiting the same degree of power suppression after the stimulation onset. Thus, we can conclude that the analysis results using Wavelet Transform, ERS-ELD, complex demodulation, and Hilbert Transform are very similar to each other. With this, I will end the comparison demonstration and go back to the slides. Now, let me summarize the characteristics of the 5 spectral analysis methods that we have discussed. The first spectral analysis we have discussed is the Fast Fourier Transform. It is a frequency analysis method that tells how much of a signal is contained in the total period of observation. The mapping from time domain to frequency domain is done by computing the similarity between the signal and the sine waves of different frequencies. When performing FFT, the assumption on the quasi-stationarity of the signal must be taken care of. Besides that, the desired frequency resolution should be achieved by either adjusting the sampling frequency or the segment length. The output of FFT is a complex number in which the power and phase can be extracted. Overall, FFT gives a perfect frequency representation of a time domain signal, but missing time information. The second method that we have learned today is the Wavelet Transform. It is a time-resolved frequency decomposition method used for localizing changes in the frequency characteristics of a signal over time. Like FFT, wavelet transform is done by quantifying the similarity between the signal and the wavelet of different center frequencies. This generates one wavelet coefficient per frequency and time point, resulting in a time-frequency representation. Due to time-frequency trade-offs, it is important to know the minimum frequency of interest that you are going to investigate. The following three methods, ERS, ELD, Hilbert Transform, and complex demodulations, are rather similar to each other. To ensure the resulting output can be well interpreted, these methods require a bandpass filtering and a prior information of the frequency band of interest, and they produce an output of a time-resolved signal. Now, you are informed with the differences and similarities of the presented spectral analysis methods. To give you some more ideas on using these methods, I would like to show you some exemplary use cases. These use cases are taken from research publications, and I have collected these papers and attached them in the literature and reference slide. In a study, FFT was used to assess the impact of gate skews in direct versus averted gates by evaluating the power of the mu rhythm. It was reported that the mu rhythm was suppressed upon movement observation. In the same study, wavelet analysis was performed to validate the choice of the mu band and explore the timing of mu suppression. According to the authors, mu suppression was confirmed maxima between the 8 to 13 Hz frequency range and was evident for approximately the entire duration of the trial. An example of ERS-LD use case was reported in a study of neural processes underlying programming experience. Task-related alpha and theta band power changes in different reasoning tasks were computed. In one study, Hilbert Transform was used to extract the instantaneous phase of the theta band and instantaneous amplitude of the low gamma band from local field potential. The aim was to measure phase amplitude coupling between two frequency ranges of interest. Lastly, in another study, complex demodulation was used to extract instantaneous power in the slip spindle band to facilitate the automatic detection of spindle events between individuals. Having performed your spectral analysis, you are likely to extract quantitative measures for further statistical analysis. Let me give you a comprehensive list of export options. If you intend to have the entire output, that is all data points, you can use the generic data export module, which works on time, frequency, and time frequency domains. For other types of exports, you may refer to the table below, where I have listed down the relevant options based on either the output is an average or single trie, and either it is in time, a frequency, or time frequency domain. Note that the options shown in green are the analyzer export options, and you can find them in the Export tab in Analyzer, while the options highlighted in blue are the solutions. They are part of the All Analyzer Solution package. If you don't have it yet, you may download it from our website. With this information, I'm sure you will be able to find options that suit to your needs, and if you need further assistance, you are always welcome to contact us. And with this, I finish my presentation, and we now have some time for open questions.
Excellent. Thank you very much, Yinfen, for such a nice overview. So just a short reminder to our audience, should you have any open questions, please feel free to use the questions pane, and please make sure also to send it to a staff. So far, we have been already addressing a lot of questions, and we have also compiled a couple of them that can be very well addressed by our speaker. So Yinfen, I will now pass over those questions to you, all right, so that you can proceed further. And let me read the first question to you. It's about the Welsh method. I'm wondering how this method differs from FFT, and how can I achieve Welsh's method in Analyzer?
Thank you, Kidis, for the questions. This is a very good questions and very interesting. In fact, FFT and Welsh methods are very similar. I would say that. So the only difference between these two methods is in FFT, the segments are not overlapped, whereas in Welsh method, the data is split into overlap segments. There is no dedicated module designed for this method in Analyzer, but we do offer a workaround to perform this spectral analysis. So let me go to Analyzer and show you the steps. So let me minimize the comparison of analysis methods and expand the resting state data. So as I said just now, the only difference between these two methods, FFT and Welsh method, it's only the difference in terms of the segments, how the segments are created. So the first step is to create these overlap segments. So we can do it in the segmentation eyes open at on the first segmented data here. So we call segmentation. It's under segment analysis and function group. So instead of create a new segment based on the marker, but now we create the segment, divide the data into equal size segments. So here we will need to define the size of the segments. So I will again use the length of 2 seconds. So the question now is the overlap segments here. So there is actually no rules or strict guidelines for how much overlap should be used, but usually an overlap of 50% is used. So for 2 second segment, so that means 1 second overlap will be used. So let me click next. So I will not create a separate data set for each new segment. So I will click finish. So now this is the segmentation on that perform on the overlaps. So you will observe that the segment number of segments now will be more due to overlaps. So after that, so we will, for the Welsh method, after that, it's same as FFT. So on top of the segmentation node, we apply the FFT. And and for the Welsh method, normally it is calculated the power density. So the power density options will be selected. And usually the Hamming window is used. And 100% of the window length will be used for the corresponding 50% overlap that we have used just now. So we will click. I will click OK. So this is actually the FFT performed on the overlap segments. And again, same as the FFT, we will perform to improve the signal to noise ratio. We will perform the average on this spectrum. So under result, under here, we will perform the averaging again, and to get the final spectrum. So this is the Welch method result. So let me take a look at the OZ. So the Welch method is performed now. I hope you are able to perform the Welch method now in Analyzer. So let me see the next question. Let me check the incoming questions. Okay, so the next question sounds like this. You mentioned that the signal should be a narrow band signal for ERS, LD, complex demodulation, and Hilbert transform. I'm wondering how narrow shall the band be? So this is a very good question. The answer to this question is I would say there's no fixed answer to this question, but in general, as what I already mentioned in the talk, to obtain a better estimation of the amplitude of power envelope of the frequency that you are interested in, which is the carrier frequency, the frequency band of the interest should be as narrow as possible. This is because all the frequencies in that frequency band will contribute to the final results. So as what I already mentioned in the talk, that the frequency of interest should be known a priori. So for example, if you are expecting a strong modulation of 14 Hertz due to your experimental paradigm, you could specify a spectral bandwidth to be 4 Hertz. That means it will be from 12 to 16 Hertz as your narrow band that correspond to the low beta waves. Or if the spectral changes are expected in the frequency of 25 Hertz, you could specify a frequency band of interest from 20 to 30 Hertz. In my opinion, a 10 Hertz spectral bandwidth would still be fine and acceptable to obtain a reasonable result. However, I would recommend performing some literature reviews in your research area to get some more ideas on the commonly used spectral bandwidth, that is the frequency ranges used by other researchers in their analysis, so that your results are comparable to theirs. I hope I answered your question. So let me check the next questions. Okay, here comes the next question. I am interested in short time Fourier transform, another time frequency decomposition method. Is it possible to compute short time Fourier transform in Analyzer? Okay, thank you for the question. So this question is closely related to the first question, which on Welch method. So actually, when we were computing the Welch method, without knowing that, we actually already computed the short time Fourier, the so-called short time Fourier transform, because the short time Fourier transform is also usually performed on the overlap segments. So this node, FFT node here, actually is a short time Fourier transform. So that means short time Fourier transform is just the Welch method without an averaging the spectrum of all the segments. So we can see short time Fourier transform actually is a time and time frequency representation. That means the x-axis will be the time. So the segments here, if you scroll, it represents the time information, and the y-axis, which is represents the frequencies. So if we plot them along the x-axis and preserve the changes in dynamic over the time, so that's the short time Fourier transform. Unfortunately, that in Analyzer we do not have this function to create a time frequency visualization from the analysis. So if you want to have such a visualization, we recommend you to export the data to external software, for example, MATLAB, to plot the time frequency representation. I hope I answered your question. So let me check what is the next question? Okay. I will pause now, receiving further questions due to time constraints. I hope we have covered most of your questions. If we were not able to address them all, please feel free to send your questions to our email account via support@brainproducts.com. Let me go back to the slides and give you some concluding remarks to recap what we have discussed in today's webinar. The important take-home messages are the following. FFT provides an adequate representation of the EEG data in the frequency domain. It can be applied as long as the underlying oscillatory patterns are quasi-stationary. For instance, alpha power during eyes closed, eyes open conditions. However, EEG signals are often not stationary and change over time. To investigate the spectral dynamics of such signal across time, time frequency analysis methods are used. In our demonstrations of applying the spectral analysis methods on the visual evoked potential data, we show that Wavelet Transform, ERSLD, complex demodulation, and Hilbert Transform yield similar results. So if you intend to study broadband activity in your signal, or you are not sure which frequencies will be modulated by your experiment paradigm, it is preferred to perform wavelet analysis. This allows you to explore the spectrum of multiple frequency bands in parallel. On the other hand, if you are interested in investigating the spectral dynamics across time in a specific band, ERSLD, Hilbert Transform, and complex demodulation will be the options to go for. Please note that these methods require a bandpass filter prior to their operations to confine the signal into a narrowband signal that consists of the frequency band of interest. I hope you enjoy my presentation and we are confident on selecting the suitable analysis method to use for your data analysis in solving your research questions. Additional materials from our site. First is our user manual, where you will find very useful theoretical concepts as well as detailed information on the mentioned spectral analysis modules in their dedicated chapters. Second is our webinar channel, where you can access the video of the FFT, wavelets, and connectivity webinars. And in order to have more information about export options, check out this very useful press release article, which will give you a quick grasp about the different export options that Analyzer offers. For further readings, we would also recommend these textbooks where you can get quite a nice introduction on spectral analysis and in general about EEG signal processing methods. And of course, a good starting point is always to check for publications related to your field of research. Lastly, I would like to invite you to read some literature papers listed here. These papers give good insights into the spectral analysis methods that we have discussed today, especially the last two papers discuss the comparison of the methods. With this, I would like to thank you for your attention. You will soon receive a follow-up email with a link to the PDF slides and a video recording of the webinar. Please do contact us at support@brainproducts.com if you have more questions about this webinar or other topics around Analyzer. Thanks again for watching this webinar. On behalf of the whole Brain Products family, I wish you all good luck with your data analysis and happy spectral decomposition. I will see you around in our next webinar. Bye-bye.